one strain from various habits, including freshwater, lining, terrestrial, and coastal regions. Thank you, Lukasz.
So very happy that this ParAqua collaboration brings Lukasz here. Thanks. Just share the presentation. The point.
Okay. Okay. Thank you for invitation. And I'm gonna talk about the isolation of and cultivation of cyanobacteria, mainly filamentous cyanobacteria, because this is my part of work. So the filamentous cyanobacteria are easier to isolate than, for example, cyanobacteria. But I hope this presentation and the practical part for some of you will be fruitful. So what about cyanobacteria? Some basic words. We have different shapes and forms of cyanobacteria, from the single celled cyanobacteria, which can also form colonies. They can produce gelatinous sheets, and they can exist in such colonies, for example, microcystis. But a lot of cyanobacteria is exhibiting filamentous form, and they are also quite difficult sometimes to isolate because of several reasons. We can find very large fascicles of, for example, Aphanisomanophlosaceae filaments. They can reach the size even 2 or 3 centimeters in the nature. But after let's say 2 or 3 years of cultivation in the lab, this special unique feature of Aphanisomanophlosaceae can disappear. Why? Because we, during the isolation of cyanobacteria from nature and their cultivation in the lab, we can eliminate the threats for them. So we can eliminate, we want to eliminate everything which is immersed in water, like grazers, for example. So what is the reason to keep such adaptive feature on when the grazers are absent? They can be found, these organisms are fantastic. They can be found in different types of habitats. Often they are first bioengineers in the ecosystems, in nutrient polluted ecosystems, aquatic ecosystems. We can see the problems they are doing, the blooms, of course, often toxic, but they can also be found in extreme habitats. Here we have the artificial pond in Poland, which bottom is overgrown by some organisms, like on this movie is visible, but isolation of the organisms from these mats on the bottom showed us that cyanobacteria, filamentous cyanobacteria, can be found there, and the temperature of the water in this artificial pond is near the outlet of geothermal water, around 50 or 55 Celsius degrees. But our laboratory work, because we wanted to see what is the thermal optimum for this geothermal cyanobacteria for several strains. So we found that this is around 26. Of course, we have the numerical data that can prove it, but also the photos conducted after 12 days of incubation and 21 days quite clear show at which temperature the growth is the highest. And here we have the culture of this cyanobacterium. This is the aggregated cyanobacterial mat that were, by shaking, this mat was separated from the bottom, and such interesting shape was found. Here we have the bottom overgrown by one strain we successfully isolated from this geothermal pond. But we can find also extreme cyanobacteria in extremely cold environments. And here you have the short movie on which you can see the mud overgrowing the stone during the expedition in Pyrenees glaciers, because we also isolated the material from extremely cold habitats. And we successfully isolated some phylamentous strains. We also stained them with the Alcian blue, and I included those photos to show you the exopolysaccharides that a lot of cyanobacteria can produce, especially in extreme habitats. The production of exopolysaccharides can protect them against heat stress, for example, but this is not only one reason why cyanobacteria produce exopolysaccharides, but production of exopolysaccharides can make the isolation process of cyanobacteria and our desire to get the pure azinic culture also very, very difficult, because bacteria, because the exopolysaccharides have the gluing properties, so bacteria can easily be binded in the exopolysaccharide, and during the isolation process, they can be transferred together with our target specimen to the culture vessel. And the bacteria, heterotrophic bacteria, can quickly overgrow the culture. Therefore, this process is quite difficult, time-consuming, and we have to spend a lot of time for isolation. Yeah, here you have one culture with small dots, maybe they are visible. The culture contaminated with bacteria, and on the right side, we applied the several mixed techniques for isolation to eliminate as much as possible the microbiota of the cyanobacterium. Here's the Plantotrix agardi case. Okay, so that was the short introduction, but the strains have own power for us. We can use the strains to analyze, to find the techniques, methods to prevent, for example, blooms. We can study the reaction of organisms to different ways to minimize their negative effects on the environment. But they have also great potential on the market. So we can isolate some molecules from cyanobacteria, and also cyanobacteria are used for production of biofuels. Here you can see that cyanobacterial scum never looked so good like on this. We can also isolate the pigments, phycoerythrin, for example, which are very, very expensive on the market. Also, some cyanobacteria can produce resting cells, akinetes, and produce especially heterocytes, which enable them to fix the atmospheric nitrogen. And heterocytose forms can sometimes be used as biofertilizers for example rice cultivation. So the potential of strains is very high, and we are on the good way to be to make the business with them. But let's go to the main topic, strain isolation of filamentous forms. Here you can see the basic equipment we need. We should do it under sterile conditions, under the laminar airflow cabinet, which is the best. If we have the big one, we can mount the inverted microscope inside and try to make the process more sterile, and it depends on our demands, our purposes. If we really want to obtain the acidic culture, we have to take care about some nuances like this, mounting the inverted microscope inside, and take care about the sterile conditions for the whole time. So we need a lot of glass, sterilizable glass, plastic plaster, glass plaster pipettes, bottles. We have different types of equipment we could use, but the basic thing is that this equipment should be sterilizable. And we need very good is inverted microscope to use. We can work comfortably using this type of instrument. And the good option is to have the camera, so we can save the information about the shape, about the special features of cyanobacteria at the isolation, at the beginning process of isolation, and we could then compare some initial results from the first isolations with the results after several years of cultivation of a given strain under laboratory conditions, to see how the laboratory cultivation affected the strain. So very basic method is dilution technique, serial dilution technique. We could apply it and serially dilute the water sample, but this method cannot. By this method, we are unable to obtain the culture without main bacteria, without heterotrophic bacteria, because they can easily overgrow the culture. We can also use the Petri dishes and the isolate cyanobacteria on agar. A lot of cyanobacteria can grow positively on agar. A lot of cyanobacteria have the benthic behavior, but also some planktic strains can grow on agar too for some time. Here you have the filaments of Planktothrix on the agar, and also, sorry for the previous name, this is now Raphidiosis, according to the modern taxonomy. So even heterocytose form can be cultured on the solid, semi-solid substrate. So the strain isolation main principle. This is the method which I usually use in my work. So I want to pick up the single specimen, my target specimen from the phytoplankton sample, let's say, and then I have to transfer this specimen to the new drop of the medium or distilled water during the isolation process. It depends on you, and after such several dozens steps, we have a chance to keep still this specimen in the subsequent drops of the medium. And if there is no other organisms, microorganisms around, we could start the cultivation of our target isolate. Yes, regarding the microcapillary preparation, the best is to use the thinnest as possible. We can buy some microcapillaries in the lab shops, but you can, if your budget is limited, you can prepare the microcapillaries yourself. And so here you can see the short movie that I recorded, how to prepare the microcapillars of different sizes.
You look like your hands are. Did you put the hand on the fire so you are super thermo-resistant?
Yeah.
It's a fantastic video.
So we have to heat the part of the glass paster pipette. I recommend to heat the middle because we can obtain from one paster pipette, we can obtain two, not only one. Yes, we can reduce the costs as much as possible. So after a few minutes, the glass will become more plastic, and then we have to elongate the pipette, and it's good to have the fire protective gloves in one hand, and the scissor is good to keep the second part of the paster pipette to stabilize during the elongation process. So in this way, we can obtain several microcapillaries, and dependently on our target organisms, if they are really, really small, not so small like cyanobacteria, yeah, but if they are very, very thin, we could have a greater chance to pick a very small volume of the medium with our target specimen, and only few other contaminants or other organisms. Yeah, and after several applications of the transfer of the specimen through the drops of media, we can have a chance to get the quite good isolate. So here you can see that we can prepare different sizes, different length for the microcapillars, and now it's good to be angry because we can easily separate the glass, break the glass in particular parts, and voila, we have several microcapillars. It's good to have the length of the microcapillar of 20 centimeters, because not too short, not shorter, because we want to work comfortably together with the inverted microscope. If the microcapillar is too short, we have to dance with the microscope, and it's very difficult to work for the longer time. Yeah, so if we have the very, very small organisms, we can use the small microcapillars. During this process, we have to use our mouth to blow out the air during the isolation. But if you are worried about you can you can use the peristaltic pump and add the pipe, connect with your microcapillar like this, because this end is has the larger diameter, so you can easily mount the two parts together, switch on the peristaltic pump, and your target specimen will go semi-automatically to the drop of the medium. So later you will have a chance to prepare one if possible. So at the beginning, it's at the beginning when we have our fresh sample from the nature, it's good to make not a big shock for our organisms. So it's recommended to use the filtered environmental water as a first stage of cultivation of our isolates. So this filtered environmental water reflects to the natural condition from which we obtained the sample for isolation. Then after, in case of successfully growing isolates, we can add more and more artificial medium to make the transition to the laboratory conditions. Actually, we have different types of media we could use, but if you want, you can prepare the medium recipe yourself. Yeah, in most of the culture collections, this is only one, but you can find another recipes in other types of culture collections. So there are a list of media, freshwater media, saltwater media, in which you can find the protocol, how to prepare the medium, and also the concentrations of particular stocks, particular compounds that are necessary to prepare the medium. You can also buy the medium from the culture collection directly if your money, if you have a lot of money and you don't know what to do with. Okay, during the isolation process, you can apply some additional steps. For example, you could apply purification of the strains with many types of compounds. For example, you can apply the germanium dioxide to, if you want to eliminate the diatoms. So sorry, Lukasz, for this information. And you can also use the solution of the medium or the distilled water during the isolation process. The medium could be enhanced with the compound, and you can bath the isolate, your target specimen, through the drops of the medium with the compound or with the antibiotics to eliminate as much as possible the bacterial contaminations. As I mentioned earlier, cyanobacteria can produce the exopolysaccharides, and here you can see the photo that bacteria can, heterotrophic bacteria can easily be attached to the surface. Some cells like heterocytes in some cyanobacteria are very tasty for contaminations for other microorganisms, which can make the process more and more difficult. So it's good to explore our sample, fresh sample, to see the filaments that are free of the attached organisms at the beginning. Also, we can try to find the resting cells like akinets and try to isolate the akinets as a single cells. Yeah, the akinets have a greater chance to survive the treatments with purification with antibiotics. So here you can see the microcapillar and the way how the team from the Lithuania obtained the culture of Aphanizomenon flossacque. So they relied on single akinets present on the bottom. And applied some chemical treatments to reduce, to exclude the bacterial contaminations. Regarding the straight purification, we also have the different types of antibiotics we could apply. It depends, of course, of our purposes and our ways. So very efficient is imipenem or tianam. We can find a lot of papers showing its power. Some cyanobacteria, like described in this paper here, they use the 4-medium and analyzed 2 different types of compounds. So 4-medium, 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 4-medi to different antibiotics. So for medium can also produce a lot of exopolysaccharides, and the production of exopolysaccharides can protect cyanobacteria from the presence of the antibiotic. The antibiotic may not efficiently penetrate the surrounding of the filament. So in case of mucilaginous producing cyanobacteria, it could be very difficult. Regarding the azinic cultures, it's very hard. Personally, I think it's probably impossible to get maybe not to get to mine for a long time the azinic culture, but it depends of our purposes. For the biotechnological applications, we have to take care about the quality of the product. So in this case, the azinicity is very important. But we should know that the elimination of the heterotrophic bacteria, some contaminations, can affect the culture condition, the growth of our target organisms of cyanobacteria, and some strains can exhibit very weak growth if they are free of microbiota, additional microbiota. And this is very expensive and time-consuming because it's not enough to obtain the azinic culture, and this culture may not be for the whole time in the azinic form. So routine, frequent checks are necessary, which is very expensive. Also, regarding the culturing of cyanobacteria, we can apply different methods. The most common for store of cyanobacteria is batch culturing in the bottles. We can use different types of bottles, plastic bottles, glass bottles. Also, we can create the continuous culture system with the chemostats and the peristaltic pump. Here we have the medium, and the medium is can be continuously transferred to the our culture. And the excess volume is moved to the outflow bottle. So this method is very good to design, to conduct some experiments with cyanobacteria. But for standard cultivation, it's very good to use the batch culture technique. And if our collection, if we were super happy with and super successful with isolation of cyanobacteria, and the amount of strains is very high, we should think how to reduce our time spending on taking care about the cultures. So in this case, the batch culturing and the reduction of the size of bottles, the volume of the media is very crucial step. So I use the small 20 milliliter bottles for cultivation, and I'm responsible for the whole process. And it's manageable by one person when the amount of culture is around up to 200. Yeah. Assuming that I have also another types of work to do at the university. But the reduction of the size is in this case is very is recommended, because we can also reduce the area in the lab for our cultures. Yeah, here you have the movie with some polar strains that can grow very slowly in the conditions reflecting their natural conditions for growth in the nature. Yeah, and here you have some photos how our cultivation system at the university looks. And yeah,
Wow.
larger collections. This is not from my lab, but from the Czech lab at Professor Komarek lab, because I spent there some nice time to learn with isolation. But you can see that you can arrange, you can reduce the space in the lab for cultivation of cyanobacteria, so you can prepare the cultures on the wall. Yeah, so still you have a lot of space for liquid, the cultures in the liquid media. If your culture collection is super big, you have to think about cryopreservation, and also some cyanobacteria, some strains can survive this process. Worth to note is that some of them can produce akinets, so the population can renew from the akinets after such treatment. But this is not universal, and the strains can considerably differ in their ability to survive cryopreservation. Regarding the literature, it's very good book to see, to explore the techniques, not only those that I simply mentioned during my presentation, but there you can find a lot of information about, in general, algal culturing techniques, because these techniques are very common, not only for cyanobacteria, but they can be applied also to another microalgae, another algal organisms. Okay. Thank you very much. And maybe some questions, then we could switch to the practical.
Just an observation. It is, in my opinion, it is not so necessary to have arsenic culture.
Yeah.
First, from the genetic point of view, you can do now analysis sequencing also with bacteria, because you can distinguish. For example, for my culture, I have cultures that are not arsenic, and we completely sequence all the genome. This is possible. So first. Second, I think that for ecological study, it's important also to know the microbiome because of the interaction. I agree with you that for some reason, from the product of some, I don't know, for some practical reason, you can have the arsenic culture, but I suggest to work with non-arsenic, knowing who is there.
Mm-hmm.
So this is my,
Yeah.
it's only.
Thank you. My cultures are also non-arsenic because
Difficult.
it's very difficult. I tried because I was curious if it's possible during the isolation. So I applied the rinsing the specimens through the antibiotic solutions, and the isolates usually couldn't survive. Even the short rinsing, the transfer, they transferred through the drops of the medium with antibiotics, and we don't have conditions under laboratory conditions. We don't have the equipment which should be necessary to work with the azenic cultures. So my cultures are also non-azenic, and I agree with you completely that why to eliminate the organisms, the mutual organisms that can benefit.
Yeah, they are important because they work together. And for some experiment, you can use some technique like nanosynths to know who is doing what.
Mm-hmm.
So if you are bacteria and cyanobacteria, and you want to know some process, some metabolism, you can do this technique and know who is doing this process. So it is not necessary to keep it on.
Yeah, I agree.
So this is just my thinking.
Okay, any other question?
Thank you for your presentation. I wanted to ask if you ever extracted the APS from the cyanobacteria.
Not yet. I only detected, but I wondered about the techniques, and there are some methods we could apply centrifugation, and the exopolysaccharides can be at the bottom. So then
But you
we
would
have
target only the exopolysaccharide and not the other polymeric substances of the EBS?
could you repeat?
Would you target only the exopolysaccharide or also the other exopolymeric?
I was focused on the cyanobacteria, yeah, not on the exopolysaccharide analysis in general.
The last one, then we go to the left.
I saw you doing some filtering of the name of the what's the biggest volume that you get?
The biggest volume
Can you repeat from the public also?
for the cultivation of the Aphanizomenon flosaccharide, the biggest volume of the medium, yeah?
Yeah, but how much of good scale of that filter? So some volumes of bubbling there, you got alcohol bubbling.
Usually my cultures are not bubbling, but I have the shaker, automatic shaker. So I have a timer. So every 1 hour, the culture is mixed for 15 minutes. I prevent bubbling because it's a lot of work to do with the pipes. Yeah, so it's easier to put the culture, because in case of large amount of strains, yeah, I can easily put the plate with 25 cultures if I have 100, so I can put all of them on the platform, and it's quickly.
You never try to scale it up.
No.
Okay, so because Lukasz will be here with us, so it's I think it's a good time to do something more practical, and yeah, I asked also Laura and no, nothing. How are you? How are you doing? It's okay.
Yeah.
So
Okay,
thank
so,
you.
I prepared some. I brought some my material, so I have some cultures.
Thank you for that.
Yeah.
Yes, thanks a
So
lot.
if you want, you can try to make your first capillar, microcapillar, if you want. Then we could start the simple experience with that. Cyanobacteria. Yeah, that's all. Enjoy. So I have the very basic burner of that. Okay, first problem. That is
I think we have the Richard C.
Me lo ricordo pure io.
Io sono stata lì. Hai visto che c'è?
Senti, io non so gestionare questo.
thanks for isolation.